This podcast is brought to you by the School of Advanced Study, University of London.
So all I want to mention is the context from our end, which is that this actually originates for us from an ERC project that I'm leading on. It's called a process philosophy for contemporary biology, which is very roughly is a project exploring the thesis that the long tradition of substance ontology has been a disaster for understanding biology. Now, obviously, I put this in perhaps a slightly contentious way, and obviously it encourages us to engage in dialogue with people in metaphysics who perhaps wouldn't agree entirely with that way of thinking of things. Some of them, I hope, are in this room. But at any rate, this project is probably sort of an equal mixture of exploring a process-focused biology and looking at a range of background philosophical issues that come up in trying to make sense of a more thoroughgoing process biology. That's what obviously one of the areas that we got interested in through this was identity, and even the identity of biological systems like organisms, and even organisms like us. So personal identity, and in particular, I became interested in this when I heard a rumor of this idea of animalism that said that there was a biological approach to the problem of personal identity. One of the postdocs on this project, and Sophie, is a sort of in-house metaphysics person and has a particular interest in these kinds of issues. So she saw this call, proposed that we put in the proposal that Ophelia just mentioned, and as they say, the rest is history. So I think perhaps at this point, I should just hand over to Anne-Sophie and let her say something about the conception we had of this conference.
Dear Ophelia, dear speakers, colleagues, dear visitors, also some words from me. It's a great pleasure for me to welcome you all to this conference, and I feel deeply honored by the official opening which you gave, and thank you, John, also for introducing us to the intellectual background in which the topic of this conference is situated. Now, it was last summer on a lazy morning that I stumbled by chance upon the Institute's annual conference call. And the text of the announcement said proposals were sought for a conference on a, quote, topic related to the notion of identity, broadly construed. And I immediately thought, that's it. I will organize a conference on biological identity which brings together metaphysicians and philosophers of biology. Now, I don't want to talk here about personal motives linked to the fact that after years of thinking about identity from a metaphysical armchair, I was lucky enough to get appointed in John's project to continue doing the same in a more down-to-the-earth manner. I rather would like to bring to your attention why I think it is simply overdue to have an exchange on biological identity across the boundaries of metaphysics and the philosophy of biology. Now, first of all, as a matter of fact, there are currently vivid debates on biological identity in both disciplines. So metaphysicians discuss it in the context of the question of material constitution, that is the question under what conditions does something compose something else, as well as in the context of the question of persistence, under what conditions does something persist through time. Now, philosophers of biology discuss biological identity with regard to recent biological studies in symbiosis, the evolution of multicellular organisms, ecology, immunology, socially organized life forms, epigenetics, systems biology, and ecological, just to name the most important fields. Though the contexts are clearly different, the thing metaphysicians and philosophers of biology are talking about is the same: the synchronic and diachronic identity of organisms. This alone provides sufficient grounds, I believe, for making efforts to merge the so far separate events, at least partially, so as to increase the knowledge of both sides and to deepen our general understanding of biological identity. There is, however, a second, more specific reason to do so, and this is that the tendencies of thought within both discourses actually run counter to one another. So while metaphysicians invoke biological conceptions of identity within the context of information for the main purpose of securing identity against skeptical views, philosophers of biology have become more and more suspicious about biological identity following the latest discoveries in biology about the nature of organisms, especially concerning their symbiotic, dynamic, and environment-dependent character. So more directly put, while metaphysicians welcome biological identity as an attractive or even the only possible solution to the metaphysical puzzles of persistence and material constitution, for philosophers of biology, biological identity is the puzzle itself. So this situation clearly calls for a more specific dialogue between metaphysicians and philosophers of biology about the issue. Is there such a thing as biological identity? What does biological identity consist in? What are the empirical facts? How do we best understand these facts metaphysically? What does follow from this for the metaphysical questions of persistence and material constitution? And what can philosophers of biology learn from metaphysicians for their own thinking on biological identity? So the aim of this conference is to initiate such a dialogue, and I'm more than pleased that all these renowned experts sitting here, our speakers, have accepted our invitation and have traveled to London for the purpose of sharing their views on biological identity with us. And I'm also more than happy that others too have found their way here in order to participate in the discussion. So again, welcome to everybody and thank you for coming. So a few words on the organizational methods. We will have coffee and lunch served outside in the corridor space. So you're welcome, as understood, to have your break there or also take food into the room. We will have a conference dinner tonight, starting at 6:30. It's very close to location. I guess the most reasonable thing is just to go and walk directly for anybody who wants to go down their own place. The address stated in this little booklet, which you find on the last page. And finally, it is my honorable duty to give thanks to the people and institutions which made this conference possible. The first to be named is, of course, the Institute of Philosophy, part of the School of Advanced Study, so which awarded us the grant with the highest possible sum. So we're really grateful for your generosity. And secondly, we also owe thanks to the British Society for Philosophy of Science, who gave another substantial contribution. And thirdly, we are indebted to the European Research Council, which will cover the remaining costs by our already existing research project. I also would like to thank Richard Somerville for the professional support in organizing this conference. It's really helpful. And I would also like to mention our secretary in Exeter, Ms. Chibon, who was really helpful with bookings, calculations, and all the rest. I wish us all an exciting conference, and thank you very much. And our first speaker is Alvaro Moreno, who will talk about what makes a connection of cells and integrated individuality.
So, thank you all for
this kind of invitation to the organizers. Very happy to be here, and especially really excited with the topic, because the topic is one of the most fascinating one in philosophy of biology current days, in my humble opinion, because it addresses so many things, so many questions that really deserve a strong and interdisciplinary discussion. Okay, so my talk is organized as follows. First, I will introduce the how I understand, which is the main point of the discussion. So addressing the question of individuality. Of course, I know that biological identity could be addressed in different ways, but why I have chosen this one, the question of individuality. So in this part, I will basically try to understand which are the main concepts that define what is biological individuality, and how is the minimal form in which biological individuality could appear, which are the implications. Then how this minimal form of biological individuality, how by its own nature creates collective systems, and these collective systems made of different forms of minimal individualities raises many puzzles, many different possibilities of combination, and that's why I will address in the 4th point the puzzles of the concept of collective individuality, why some systems deserve the term of composite or collective individuality, and why maybe others not required to be characterized as such. And then the next point which I will address is which is the organizational background for understanding these composite individualities, which is the concept of I will develop of functional integration, then the implication of this concept, and finally the concluding remarks. So, just for an intuitive perspective of what we see in the very diverse and incredibly rich technology of the biological domain, is that we have the idea that there are some cohesive entities that we call individuals, individual living entities, that show certain interesting properties, and that usually, but not uniquely, are equated with what the biologists call organisms. Now, first of all, what I mean by individuality, I know that there are a very strong debate about what is individuality. There are many other concepts that are not organisms that are qualified as individuals, species, genes, etc. But I will use the term individuality for referring to systems or entities that distinguishes themselves, it's important, from other neighboring systems or entities by themselves. And the point is that these individualities, these entities, show certain capacities which, in my view, and I will try to explain all this claim during my talk, concentrate the most interesting capacities that the evolution of life has produced: functions, agency, cognition. So the point is that my claim is that we cannot understand how this through the evolution, how biological systems have been able to produce more complex and new and interesting capacities without addressing the question of individuality. And an organism-centered view, which is my claim, is fundamental because through this view, and only through this view, we can define where selective forces operate, define on which basis or what specify ecological interactions. By that I mean that when we define ecology, we implicitly take for granted that there are some items of biodiversity, and these items define the interactions. Now, just for giving an example, suppose that we are talking about a termite that defines some interactions. Now we take for granted that this is the termite as a whole that defines this interaction through his agency. And no, for instance, their biota in his goods explain the appearance of increasingly complex bodies. And finally, explain the emergence of new interactive capacities, complex agency, and cognition. And for these reasons, I claim that the focusing on the individuality and the minimal form of individuality is organism is very important. So then I will start with the basic form of individuality, of organismality, which is the cell. The cell is broadly speaking, a cohesive, topologically close, self-maintaining organization, which is the source of biological individuality. And now I will distinguish two complementary dimensions or levels of the description of the cell. One is, recall the constitutive dimension or level. From this perspective, the cell is a recursive network of reactions maintaining in far from equilibrium condition through a set of functional constraints, like the enzymes, that are generating within the network. And I think that maybe another talk will address this question more carefully. Yours? And the second point is that this network produces a selectively permeable compartment, which is a functional constraint also, but that acts globally and therefore covers all the remaining functional constraints within it. And this is important because this global constraint, which is in nowadays cells quite complex, but is what in some sense integrates all the remaining functions, and also it produces this individuation that is characteristic of the cell. And just for understanding the importance of this point, let's come back and let's have a very brief view on the origin of life, the prebiotic evolution. And of course, it is important that the phenomenon of life is a multidimensional phenomenon. On the one hand, it is creating a collective and historical process of interaction, of networks, of intergenerations. And on the other hand, there is a form of embodied proto-organismic organization. Now, the point, and my point is that this latter aspect, the creation of units, self-enclosed units in far from equilibrium conditions, is the organizational core that, in a fundamental sense, is the cause of the other dimensions, and at the same time, of course, the other dimensions affect also the possibilities of long-term maintenance of these individualities. So that's why the central point is the process of self-encapsulation, because this is, on the one hand, the process of minimal form of individuation and minimal form of integration, because it's through this entanglement between the catalyst, the enzymes, and the membrane that the system becomes a whole integrated. This organization defines a minimal form of individuality, therefore. Now, at the interactive level, or dimension, the cell deploys processes modifying its environment, and such that this modification is what ensures the maintenance of the constitutive identity. So take for example the case of a chemotaxis. Since the biological identity is a far from equilibrium, is a precarious form of identity, the cell should do things in the environment sooner than later in order to keep going its organization, its identity. On the one hand, this internal organization is the source of these interactions, this form of agency, but on the other hand, in different time scales, is through these interactions that the system keeps going. That's very important. And second is that this concept called agency expands the functional control of a biological organism, and this externally expanded functional control could in turn be recruited to increase constitutive plasticity. It's by doing things in the environment, and this is what we call adaptive behavior, is a way in which the system expands its own possibilities, not only by creating new metabolic pathways and so on. It's also by doing things in the environment and modifying the conditions of the environment. And these two aspects constitute what we have called autonomous. This is what why the cell is an autonomous agent. This is just a picture illustrating these two concepts and how they are entangled, taken from a member of our group, Which are the implications? This is the second point. Now, since autonomous agents by nature deploy agency in the environment, they deploy interactions. And when these interactions provide something that for another agent becomes an input that becomes something beneficial for the other agent, this necessarily creates a network of interaction between different agents, so that these agents become complementary, functionally speaking. It's kind of illustrated like that. This is a very, very wide notion. So you have agents, and by its own nature, agents interact in the environment. But you have to realize that we are talking not only in the physical environment, but also the environment is constituted by other agents. And these other agents are related. Suppose that, for instance, some agent produces a waste, and this waste, suppose that this waste is an input, a food for another agent, and so on. And this exactly means that the minimal form of synchronic cooperation, or synchronic web, is an ecological system. An ecological system is a system in which different autonomous agents have a kind of supra-metabolic organization, in the sense that all of them create complementary food webs. And by the way, there are many arguments, I will not develop here, to think that a form of proto-ecological system would probably appear before life as we know it in a prebiotic stage. Among other things, because otherwise probably the whole system would have collapsed. Which are the implications of this perspective? Now, since autonomous agents are interdependent and opportunistic, they develop increasingly extended functional relations, like I have already mentioned, ecosystem, like colonies, like symbiosis. All of these interactive processes could become a constitutive process for a larger collective system. Now, suppose that we are changing our point of view and consider that then the system, the autonomous systems, are just parts producing a larger collective self-maintaining organization. And then, from this perspective, the former autonomous system become just parts that enter in the constitutive process or dimension of this larger collective system. When something like that happens, this collective self-maintaining system could improve the survival and occupy new niches, just for example, because they are larger, because they can do things that the individual basic entities cannot do, and display, in many occasions, some new interactive capacities. As these new interactive capacities in turn open the opportunity to new forms of successful self-maintaining associations, and so on. Just think, for example, in the case of biofilm. Practically, there are independent forms of life at the level of the basic autonomous system, which are prokaryotic world, but it's very, very common the fact that they organize together, and the biofilm is one of the most well-known form of organization. The bacteria organize together, they develop a matrix that brings together, and the interesting point is that they develop some forms of some capacities that permit that the whole system ensures a form of survival that individually cannot be achieved. Now, two things could happen here. On the one hand, well, in fact, evolution has produced many, many different types of associations. But we can see two kinds of aspects from this once we look at this collective association. On the one hand, if we look at the parts could recover, they produce autonomy when environmental conditions change. For instance, there are individual entities that associate when, for instance, the environment becomes too dry or with some stressing conditions, and then they form a collective organization that permits to survive in these different conditions. But then when the environment comes back to the more favorable conditions, then the units become again independent. The other possibility is that they lose their former independent life, but and so the process is become irreversible. But at the same time, they're still maintaining the properties of as an autonomous entity within the collective entity, and I will develop this point later. And also, finally, in certain cases, some autonomous entities could even lose their autonomy, their former autonomy, and just become a functional part. But I'm putting this in a very suspicious form, or because we will see to what point. And just allow me to discuss this point with the example of the creation of the eukaryotic cell. It's well accepted that the eukaryotic cell is a result of an association, a symbiotic association of several types of bacteria and archaea. But this former bacteria, in the case of mitochondria is the case, is not anymore something that could be qualified as an autonomous entity like a cell in a multicellular organism. Even though losing this characteristic of being to be qualified as a cell, it is not so reduced to a part that is not able to develop some of the core metabolic capacities that a cell is capable to do. So even in this extreme case, the part retain some of the functions of the minimal individuality. And looking at the collective organization, these new collective entities could display very different forms of cohesion, and not all of them could be clearly qualified or identified as full-fledged individuals. And actually, the identification of collective forms as a real full-fledged individual raises many challenges. Among other things, because instead of the case of the cells, of the minimal cells whose parts are molecules, here we are dealing with systems whose parts are already cells and with their own form of agency and without minimal form of its own normativity, and this creates many different forms of association that raises many puzzles and many different possibilities. So that the degree of individuality is something that deserves a clear discussion. I will now discuss or present very briefly just four examples of systems that present more or less interesting collective properties or identity, but that for concerning other aspects are not so, or could be discussed whether it realizes a form of strong individuality. The first example is Paramecium bursaria, which is, Paramecium is a eukaryotic unicellular organization. This cell has some cilia that produce movement, and it's full of green algae, which are prokaryotes. The algae provide the Paramecium with the food needed. They are photosynthesizing the light, and they provide to the host many advantages, mainly this one. But on the other hand, the Paramecium provides to the alga first a very homogeneous environment. They provide protection, and also they move towards light, so that the Paramecium allows to the alga to be in the adequate condition, not depending just in random possibilities to get the light. The second example, Mixotricha paradoxa, is another case of an unicellular symbiotic eukaryote. Mixotricha paradoxa is, instead of the former case, is an endosymbiont in the good of some termite. And this is a very interesting case because you look at how the membrane of this eukaryote, there are hundreds of thousands of cilia that allow the movement, and some more large rose-style flagella. These are, in fact, symbionts, prokaryotic symbionts. In fact, the cilia are spirochete, treponema spirochetes, And second, the flagella are rod-shaped, different type of bacteria, and together they allow the movement of the Mixotricha paradoxa. And in addition, within the cytoplasm, there are some mitochondria, which in fact are not mitochondria, but other type of bacteria. So this is a kind of much more interesting example of association, because there are different types of organisms that together constitute the whole system. In addition that this system enters as within another, which is the termite. The second case is the third case is the volvox. The volvox is an association or a kind of colony or whatsoever made of, could be more than almost 50,000 cells, eukaryotic cells, forming a self-reproductive, self-moving, with its own developmental process entity, that in which each cell has a pair of flagella that beat in synchrony, and that this way together they move the whole system. I'm sorry. It's depicted here, and they allow the whole sphere to move in direction of the light. And this, for example, increases the speed of the way in which the colony moves. So, by this way, by this association, the volvox is capable to do things that the individual cells cannot do. The fourth example is the termite mound. This example is different in the sense that here we are talking about a system which is made by units the units are cells, but the units here are whole metazoa, actually termites, which are quite complex entities. And the termite mound is a very complex system which includes a material nest. The material nest is made by a mixture of soil, thermal saliva, and dung, and is a very big construction with many tunnels. That is organized in you can see, for instance, just to, well, the trees give you some idea of the size, but realize that under the ground level is around 2 meters below. So it would be very, very big size construction. There are plenty of tunnels. There is a kind of organization that is a kind of chimney, that all this system is for ensuring that the temperature within the system is controlled, is cooler than in the environment. So there are a lot of termites that are working all the time for making and repairing the tunnels. So there is a place for the only one, actually there are two, the king and the queen, which are the only two parts or individuals that have the reproductive capacity of the whole system. So it's a kind of reproductive bottleneck at the level of the whole termite mound. There is also even a species of fungi that are cultivated for the production of food, some kind of food, and the termites provide the fungi with special conditions of humidity, etc. So it's a very complex organization in which the survival of the termite is dependent on how the system is collectively organized. Well, despite their functional unity, well, however, it is not clear whether these different types of examples really show the degree of integration that deserve the term of individuated organisms. For example, there are authors that claim that this last example, they call that they are superorganisms and they realize some form of individuality. Others dispute that. So to answer this question, to address this question, we should first consider the following set of questions. And I propose to follow these different possibilities or dilemmas. Cohesion. Are the parts more or less cohesive than the whole in the case of a collective system? Frontier. Is there any clear-cut global constraint or mechanism that operationally delegates the system, global system? Where does natural solution operate? On the parts or on the whole? Which organization ensures the energetic, self-maintenance reproduction, and in some cases also the development of the global system? Does the collective organization explain the appearance of novel and more complex properties? So to understand cohesive multicellular individuality as a source of the appearance of new properties and more complex capacities, my claim is it is necessary that the system show at the global level a higher degree of functional integration than that achieved in the level of the parts, in this case, the cellular level. And here I will distinguish between two forms. One is mere functional coordination. Without functional coordination, there could not be any form of collective organization, because then it would be not a viable system. But one thing is to understand that the system shows functional coordination, and the other thing is to consider that the system shows functional integration. And therefore, I will try to explain which is the difference between these two cases. The reason I claim that why strong individuality requires functional integration is because when we try to merge into a unique system a different set of autonomous entities, the forms of organization could be different. And for example, some of these entities could produce a kind of collective organization, a kind of self-organizing entity that helps some of these, that provides some advantages, but where the main form of integration remains at the level of the parts. In other cases, it's not like that. So there could be very different issues. But let me again come back to the example of the eukaryotic cell. We know that for more than 1 billion years during the evolution of life, the forms of organization, the collective forms of organization has been taken biofilms and many other forms of between bacteria. Now, however, none of these forms has achieved really revolutionary, so to speak, novelties in terms of, for example, morphology, or in terms of having the capacity to integrate together a collective organization that has the size of thousands times of the bacteria. This requires so many conditions in terms of management of the genetic information, management of the energetical processes, management of synchronization of processes, that it is not easy just by force of functional coordination, not easy at all to achieve such an integrated form of organization, composite, but capable to show really important novelties, like, for instance, exploring a universe of complex morphologies and many other possibilities. So, what is required for achieving global functional integration? First, the appearance of a hierarchical regulated organization of functions. Not many functional organizations, as I have already said. This means that the system gets organized in dynamically decoupled domains, yet functionally connected. What I mean by that is that, for instance, that the system creates, for example, spatially separated domains or temporally separated domains that behave to a certain extent autonomously, but that globally are functionally connected. So this implies that the system is organized hierarchically. These functional connections are the consequence of a dedicated subsystem or module which operates at a higher level. This means that these domains are not only a kind of self-organization. No, they are selectively controlled, they are modulated, they are sometimes suppressed and sometimes accelerated and so on by a higher level control device. This does not preclude that the system shows forms of self-organization or distributed organization. Just on the contrary, the important point is that these forms of self-organization or distributed organization are in time selectively modulated and used. All that is not the only requirement, because it's not sufficient that the system shows some regulatory centers, but that globally all these regulatory local centers should in turn be globally connected in a center or a global center of the system, which takes over and selectively organize all the functions, including many local regulatory subsystems, as I'm saying. Yet, there is a limit in this kind of top-down control. All living composite individuals are made by cells, and this means that there is a level in which the parts are taking over their own process. So it's some form of ultimate subsidiarity in the system. So this is not, the top level control is not controlling the lower level processes at the cellular metabolism level. This is something that is performed by the cells. And the last point is that the regulatory center should constitute an integrate self-generating a new form of agency, and I will address this last point, capable to ensure the maintenance and even the development of such integrated multicellular constitutive identity. So therefore, my main thesis is that it should have, for having a functional integration, a kind of symmetry between the constitutive level, the global multicellular constitutive level, and the interactive form of agency. So a condition for realizing full individuality is this symmetry. And the reason lies in the fact that since we are talking about association of agents, the form in which these associations could take many possibilities. Just for giving one of the already mentioned examples, the ball box is realizing a form of agency, a collective agency. But this form of collective agency, except for the fact that it increases the speed, does not show a qualitative kind of higher complexity than that it is displayed by the cells, the individual cells themselves. Whereas, for instance, if we take the case of an animal, we show that the animal is capable to show a full form of agency in terms of rich, diverse functionality and complexity of the agency, qualitatively different than that performed by the parts. So the implications are twofold. On the one hand, at the constitutive level, a multicellular individuality requires the combination of genetic homogeneity, because otherwise the conflicts would be catastrophic, and functional plasticity to allow that, so that the cells, on the one hand, should be genetically homogeneous, but at the same time they should differentiate in order to produce variety in terms of tissues, organs, and so on. And this requires various forms of complex epigenetic mechanisms that control cell distribution, lifetime, differentiation, location, etc., etc. And in addition, on the other hand, this is not enough, because at the same time that this specialization should be complemented with metabolic forms of tasks that the system cannot, or if doing, could be having serious trade-off problems. And this is the reason why, since the beginning, the association with a population of symbionts that perform basically, not only, but basically of the prokaryotic world, complement the metabolic task for the whole system. And of course, these the possible conflicts require that the whole system has a global monitoring system that ensures the cell maintenance, the adequate cell maintenance, which is the immune system. And at the interactive level, this complex multicellular organization in turn requires the capacity to display a rich plastic and fast form of agency. The way these two aspects are entangled is very important. This form of complex agency is what maintains and even contributes to the developing and reproduction of this complex integrated body, and conversely, this complex integrated body is the only possibility to create a metabolically coupled tissue, that is the nervous system, that supports complex forms of fast and plastic agency. Which in turn, this capacity of interaction has two sides. Not only for performing agency that through interactive processes maintains and ensures the precarious maintenance of the body identity, but also monitoring the constitutive processes by controlling the respiratory rhythm, the digestive processes, by cardiac processes, and so on. So the nervous system is also monitoring all the internal processes. And this way, in fact, we have two different systems. One is the immune system that performs a slow constitutive monitoring control, and on the other hand, we have the nervous system that performs a fast interactive sensory motor control plus a fast constitutive monitoring control. And these together, in forms that we don't know, unfortunately, the research is coming nowadays very fast, but we really don't know how these two very important systems are intrinsically and deeply related, but these are what they constitute this regulatory center, what I have called the self of the system. So finally, going to the conclusions. The emergence of a cohesive, individuated organization is a fundamental source for the complexification during the biological evolution. And this is a form of individuality which is not observer-dependent, is something that is self-created, self-produced by the system. And once a minimal form of individuality has created, this form of individuality, by its own nature, creates a network of relations and therefore tends to produce some composite, more complex form of individuals or associations, but only some of them, those that achieve what I have called functional integration, deserve or could be considered, in my view, as new forms of individuality, composite individuality.